Subscribe. Genome sequencing. The Department of Biotechnology, DBT, recently said that the exercise to sequence 10,000 Indian human genomes under the center-backed Genome India project is about two-thirds complete. About 7,000 Indian genomes have already been sequenced, of which 3,000 are available for public access by researchers. The United Kingdom, China, and the United States have launched similar programs to sequence at least 1 lakh of their population's genomes. About genome sequencing. A genome is a complete set of genetic instructions which are present in an organism in its DNA. Sequencing is the sequence of occurrences of the 4 nucleotide bases, i.e., adenine, A, cytosine, C, guanine, G, and thymine, T. The human genome is made up of over 3 billion of these genetic letters. The whole genome can't be sequenced all at once because available methods of DNA sequencing can only handle short stretches of DNA at a time. While human genomes are made of DNA, deoxyribonucleic acid, a virus genome can be made of either DNA or RNA, ribonucleic acid. Coronavirus is made of RNA. Genome sequencing is a technique that reads and interprets genetic information found within DNA or RNA. Significance of genome sequencing. Origin of epidemic. Genome sequencing helps researchers understand the arrangement of the makeup of DNA or RNA. Sequencing the genome will help us understand where the certain virus, for instance of SARS-CoV-2, came from and how it spread. Control the spread. Sequencing is useful to know and check if the strain is evolving. By knowing change in genes, it can help in preventing its spread. For instance, scientists already know the number of mutations that arise on an average in a month in the case of COVID-19, its incubation period, and the average time between cases in a chain transmission. Evolutionary studies. Global science would also benefit from genome sequencing, which would provide data useful for the mapping of the spread and migration of a range of life forms in the Old World, and thus would help in better understanding of human evolution. Determining gene-disease link. Human genome sequencing is important to establish a link between diseases and the unique genetic makeup of each individual. For instance, cardiovascular disease generally leads to heart attacks in South Asians. If such propensities can be mapped to variations across genomes, it is believed public health interventions can be targeted better. Better understanding of diseases like cancer. While genes may render some insensitive to certain drugs, genome sequencing has shown that cancer too can be understood from the viewpoint of genetics, rather than being seen as a disease of certain organs. Drug efficacy. Another advantage of genome sequencing is that information regarding drug efficacy or adverse effects of drug use can be obtained. Drugs developed in the Western world and sold in India are pricey and may not be effective on the Indian gene. Mapping of India's genetic landscape is critical for next-generation medicine. Agricultural usage. It will enhance India's scientific capabilities. Next step would be genome sequencing of crops that would help in better understanding of the genetic basis of susceptibility of crops to blights, rusts and pests. It may become possible to deter them genetically and reduce dependence on chemicals. Significance of the Genome India project. It started in January 2020 with the involvement of about 20 institutions across India and with analysis and coordination done by the Centre for Brain Research at ISK, Bangalore. The centre's Department of Biotechnology notes that the project will help unravel the genetic underpinnings of chronic diseases currently on the rise in India. India's 1.3 billion strong population consists of over 4,600 population groups, many of which are endogamous. Thus, the Indian population harbors distinct variations, with disease-causing mutations often amplified within some of these groups. Creating a database of Indian genomes allows researchers to learn about genetic variants unique to India's population groups and use that to customize drugs and therapies. Challenges in genome sequencing. Very high target. The aim was to sequence at least 5% of the samples, the minimum required to keep track of the virus variants. This has so far been only around 1%, primarily due to insufficient reagents and tools necessary to scale up the process. Low capacity, the 10 laboratories together can sequence about 30,000 samples a month, or 1,000 a day, 6 times less than what is needed to meet the target. Fund crunch, funding is being delayed repeatedly. NSEC asked for 100 crore, but it was not until March that any funding arrived and it received 70 crore. Sample collection, the healthcare system is already overstretched and this is one additional task for them to sort and package samples and RNA preparations regularly for shipping in a cold chain to sequencing centers along with recording extensive metadata to make sequence information useful. Dependence on imports, the process of genome sequencing slowed down due to the Atmanirbhar scheme which banned imports of goods worth less than 200 crore to promote local procurement. Even after the exemption, some special plastics inadvertently remained within the import ban affecting the process. International aspect, the poor progress in genome sequencing also affects India's image abroad, as all countries are required to upload data into a common global repository, called the Global Initiative on Sharing All Influenza Data, or GISAID.